So, collaborative effort, I should say. A bunch of guys got together and listened to me ramble and told me I was wrong and then told me to do this. So, don't blame me for any of this. Okay, including the rearrangement of your wondrous slides. So the real problem here is, as I complained this morning, it's terrible. We can't do all the gene therapies we want, and since small molecules exist, everybody thinks this isn't a problem. But it is a problem because small molecules are terrible in many, many important ways. So the usual caveats apply in that next to nobody sees what isn't there rather than what is there, and particularly the executives of big pharma companies. So what we want is a gene therapy platform that enables 80% of the important arbitrary combinations of functions that we want that we can't do today. And there are several paths which seem plausible in terms of actually getting there. Option one is the expensive plausible path, which is let's just make adeno-associated virus work, because if we wait 20 years, it will work. It will be able to do most of what we wanted to do because people will have crushed the cost down, just like happened to monoclonal antibodies. They will have figured out how to do repeat dosing and stop the immune system getting angry. They'll have figured out how to do the cost-effective tissue tropism issue where you can get 30 times as much AAV into random small tissue X than would normally be the case. And if you could just get those three things done, this would be pretty useful. So people are working on these things. There are some allegedly decent solutions sort of percolating through the development process. So go put together $100 million from some philanthropist who likes gene therapy, buy out their patents, buy the companies, and then release this freely for a low fixed royalty if you like money, or just freely if you like kudos, and go from there. But I do think this is an expensive one because most of these efforts are somewhat advanced, or at least interesting enough that they will be the cause of a fight to buy them out with various entities if you want to go in and do that. So option two is the low-tech option that we actually know nothing about. We folk who thought about this, there is actually a automated injection industry, funnily enough, and I know very little about it other than it exists. Is it actually possible to make a magic injection robot? One that can take the 0.1 to 0.2% mortality you get when humans do deep tissue injection in old people and convert it to an approximately 0% mortality. And my guess would be this looks an awful lot like the AAV situation in that there's going to be some work in progress that you'd want to go license or buy out or release the patents or whatever. And maybe, and if you could do this, just to be clear, if you could do this, existing gene therapy technologies today are good enough. You could pretty much get everything done you want to get done in the portfolio if only you could just inject it into the tissue you wanted without killing a modest number of people. Though I should say that killing a modest number of people seems to be a very popular goal in today's world, something we need to work on. So option number three. This is the alternative to AAV, which is let's take the state of lipid nanoparticles and DNA, RNA type delivery and just get it over the hump. Because there's a lot of interesting work in the labs. Unlike AAV, it's not further along, and we don't really understand why this is. So this really is probably a $20 million research funding exercise. Go identify the groups we know about, various labs that are working on this or claim they have something that works. Throw a few million dollars at each of them and just say, go to work, run some conferences, get people into the room and don't let them leave until they figure out what's going on. And there are just a few things we need to do here. You would need to be able to nuclear target plasmids in order to get sustained gene expression from plasmids in non-dividing cells. You need tissue-specific control of RNA expression because RNA it just expresses wherever you put it, which makes it not very helpful for some uses. You want, ideally, much better promoter control than you have. If you could just do two promoter control and have and not as logic operations, you could do most tissues with that. And you want LNPs to be a lot less toxic than they are right now, even given the vast improvement that we've seen since first generation LNPs over the last however many years it's been. And lastly, the option that I have to say, but is probably not good, is to take take the Calico type approach and throw $100 million at fundamental research to say, okay, this is all garbage, we think we can do a lot better based on something that's still in a comparatively early stage in the labs, and there's various suggestions in the list there, like nanodiamonds that people have already suggested are actually not that great, but okay, but maybe there is something there that's great and solves all these problems, and maybe you could get it to come out more quickly if you put it together a dedicated foundation to get that done. So there are people in the world who could make this happen, any of these, if they decided that they wanted gene therapy to exist badly enough. And maybe if we all went out there and talked about this a little more, and certainly the next time I make a complaint about gene therapy in the form of a PowerPoint, I will add these notes at the end in the vain hope that if I present a solution in addition to a problem, that somebody will actually do something about it for me. Fini. Happy to take questions.
So my question is about option 2. A couple of things about that. So one thing is, and you say, do we know what the cause of the mortality is? So two things. One thing is the cause of the mortality, if I had to guess, is probably anesthesia related. So I don't know if your magic robot is going to fix anesthesia risks, or infection related, which maybe a robot could be helpful for not spreading infection. But I also would question the premise of it. So if you're going after, diseases that cause people to die, is that mortality risk worth it?
It doesn't matter whether it's worth it, because the FDA's mandate is that there will be zero risk at any cost. Therefore, if you have a treatment that's going to be used in basically healthy older people, and I put quotes around healthy there, then you're not going to be allowed to go inject 10,000 people if you're going to kill 100 of them.
Well, what if they aren't healthy? What if you have surrogate markers for lifespan? And
my suspicion is, again, that you're going to have real trouble selling a 0.1% mortality solution to any condition in which the person is not going to die in the immediate future with the FDA. But this is speculation based upon my interactions with them to date. It's not quite, the FDA is not quite a magic balancing machine that perfectly trades off risk of death with risk of disease. I think it's a magic balancing machine that doesn't want to seem responsible for anybody dying.
So you should put the $100 million estimate for how much to fix that.
Yeah, it's a good question. And in terms of do we know what causes mortality? This is, if I was a wealthy person who wanted to do this, my first option would be to go talk to the researchers, send out my minions to talk to the researchers, put together a conference, learn everything I needed to so that I know as much about that as I currently do about AAV, and then start to answer that question. Because I suspect that most of the people in that industry understand what they're doing quite well.
I mean, this doesn't sound about any surgery. So if the risk is just surgery.
Yeah, if the risk is anesthesia, for example, yeah, we need a magic anesthesia robot, not a.
Or you can just argue that surgery is already accepted for many things.
Okay.
So there is a huge industry already exists in this area. Of course, there are major companies, labs, centers, initiatives, conferences exactly on that topic. But and of course, they work very, very actively on developing all these delivery tools and gene therapy, cell therapy, and so on. But let's say it works, yeah. So how would it help our kind of area? Because if the aging is kind of systemic process and we can fix a few genes, why would it be a major advance for the field?
Yes, so from my perspective, it's a major advance because certainly I'm in an area where I want this capability. And if I had it, I'd be a lot further along than I am right now in terms of removing garbage in the arteries. So there are a number of parts of, obviously a lot of, I'm a SENS-focused person, 7 deadly things, and a lot of those don't need gene therapies. You don't need a gene therapy to kill off senescent cells reasonably well. for example, but you do need gene therapies for a variety of the other items that would be viably corrected. And for every small molecule approach you see out there that is actually, useful rather than just trying to mimic calorie restriction, there is a gene therapy that would work about 300% better, because no small molecule can ever be as good as a gene therapy if the purpose of the small molecule is to manipulate a specific set of protein expression. One of the things that irks me to no end in the research community is, in general, is people will roll out a proof of concept in mice to show that the mechanism exists, and these mice are genetically edited, or they used a gene therapy to show that it works, and then they say, okay, what next? Well, let's go find a small molecule that does the same thing, except the small molecule turns out to be terrible and just good enough to get past phase 1. They get their investors their money back, and then it fails in phase 2 or phase 3, gets canned by big pharma, and that's the story of 90% of drug development, which I think would not be true if we were using gene therapy. That's probably a glib answer.
Wonderful. Thank you.
Thank you.
That's all, Scoris.